Hi everyone! For chapter 28, we will go through the anatomy of the reproductive system as well as the process in which gametes are formed. Now, the main function of the reproductive system, as we all know it, is to produce, store, nourish, and transport functional male and female reproductive cells called gametes. Now, the organs that makes up the reproductive system, either for production of the gametes itself or assist in the maturation process of the gametes itself, includes a couple things. You have gonads, which are the reproductive organs itself that produces gametes and hormones for the reproductive system to work properly. And you have ducts for receiving and transporting the gametes to their proper places. And you have accessory glands that will secrete fluids into ducts to either assist in movement of the gametes or for maturation process of the gametes. And of course, you will need external genitalia for excretion of those gamete cells. Okay. So for males, the testes are the main gonads that produce gametes, which are sperm cells. The male sex hormones, androgens, specifically testosterone, is also produced by the testes. Males' gametogenesis process is actually very different from females. One main difference is that males can make half a billion of sperm each day. Of course, that's very different from females. Thankfully, we don't have to ovulate half a billion eggs each day. That would be kind of scary. So for females, the ovaries are then the main gonads for our reproductive system. They produce gametes called oocytes, which are actually all made within the female's body when she is still in her mother's womb as a baby. And we'll talk about the process of oogenesis in a bit. Their development are just stopped at an immature stage when we talk about the development of these oocytes. Okay, so they're formed in the female's body as a baby, but it's kind of stopped at a certain stage and then continues each month, and one of those eggs will mature and will be ovulated. Now, the ovaries also secrete the female sex hormone, which is mainly estrogen. Okay, and we'll mention a couple other female sex hormones when we get to the female reproductive system. So let's start with the male's reproductive system first. We will go from where the sperm is made towards where sperm exits out of the body, anatomical wise. Most of the male's reproductive anatomy are outside of their bodies, which is again contrary to female's reproductive system, which is mostly on the inside of our bodies. The spermatic cords are the very first thing that's on the outside of the body. There are layers of connective tissues, fascia, muscles that surrounds blood vessels, nerves, lymphatics, and mainly the ductus deferens that goes from the testes. The testes actually sits in the scrotum. So this area here is the scrotum. Okay, and that's why the spermatocord, which is that section right there, is all those layers of tissues that is supposed to protect those important blood vessels, nerves, and ductus deferens. Okay. Now it will lead toward from the testicles up into the abdominal pelvic cavity, which is this area right here in the back. Okay. There's a passageway through the abdominal muscles called the inguinal canal, which is a little tiny opening around right here that will allow for the blood vessels and all those different ducts to go through. Now, even though it's supposed to be for just blood vessels, sometimes that opening in the abdominal muscles in males is why some of them can have a condition that's called inguinal hernias, where visceral tissues like intestines, mesenteries, and anything that's, sort of, flopping around inside the abdominal cavity can actually slip into that opening and cause pain. This is more likely to happen if the abdominal and groin muscles are weak and pressure was added to that area. Of course, it's an easy fix. Usually surgery can be done to repair that opening area. Okay. The scrotum are the layers that surrounds and protects the testes. So each testes are in their individual compartment. As you can see, this testicle is residing in this separate compartment, and this testicle is residing in a separate compartment. Now, this is for a good purpose, so that, if one testis gets infected, it is less likely to spread to the other testis. So at least we have one functional testis in case one does get infected with bacteria or pathogen. Now, because the testes are a very sensitive organ, there's a layer of serous membrane called tunica vaginalis that also surrounds the scrotum on the interior part to prevent friction. Okay, just like how we have serous membranes around our lungs, our heart and intestines, around our testes, there's tunica vaginalis. Okay, now the funny or sort of contradicting point about it is it kind of sounds like a female part. So make sure when you see this on the exam, if it's asked, don't think, oh, vaginalis sounds like a female part. Okay, it's actually a serous membrane that lines the scrotum of the male reproductive system to prevent friction as testes are moving and rubbing against the interior part of the scrotum. So just as we have mentioned, the testes are very important for production of sperm. Okay, this is a process that's called spermatogenesis, and this process is actually very sensitive to temperature differences. According to the textbook, sperm must be produced about 1.1 degrees Celsius. Is that exact? 1.1 Celsius, okay? Lower than body temperature, which also explains why the testes are located outside of the body instead of the inside. Because of this temperature sensitivity, the scrotum actually has the ability to contract and distend, okay, kind of like muscles. So these are made possible by two main muscles. One is called the cremaster muscle, which is a type of skeletal muscles that males can control, and the other is called Darto's muscle, which is a type of smooth muscles. Now, when the temperature around the groin area gets too warm for the testicles, the scrotum will distend and allow the testes to be situated farther away from the body to cool down. And when the temperature gets too cold for the testes, the scrotum then will contract and they will be closer to the body to keep the warmth around themselves. So that's why these two muscles functions critically for keeping the temperature inside the testicles always at homeostasis, which is, of course, our common theme throughout the entire AMP2, okay? Keeping homeostasis. So even though the testes look very simple on the outside, the process of spermatogenesis goes through many densely packed ducts inside, and sperm cells has to be lubricated with many different types of secretions from multiple glands. So we'll talk about each of these. Where do the sperm travel, where they become motile, and what kind of secretion needs to be added to the sperm in order for them to be functional. So inside the testes, they are divided into lobules, okay? Just like how we mentioned lobules in pancreas, liver, and kidney. Lobules just basically mean a section of that particular organ. And their lobules contain very important ducts called seminiferous tubules on the inside. So if we look at this particular image, right here, okay, so this is a particular one particular lobule, and inside you see these spaghetti looking noodle stuff inside. Those are the seminiferous tubules. They are very densely packed together, and that's why when you looked at a cross section of the testes under the microscope, if you remember, you see a whole bunch of circular or tubular shaped things, because those are all the tubes that are stacked and coiled on themselves as they're packed densely within the testes. If one can stretch the entire length of the seminiferous tubules out, it can be as long as half a mile. So that's how much noodles that's being packed within this tiny organ of testicles. Okay, and that's why we see these circular parts, is because we're taking a cross section of that testicle and looking at those tubes in cross section. Okay. Now, there's a specific direction in which sperm cells move and mature as they move along the way. So from the seminiferous tubules, which are all of these lobules down here, they will move on to what's called the straight tubules. And from the straight tubules, they will get to an area that's called, so here's straight tubule right there. And this net looking thing, kind of look like a mesh, those are called reti testes. And then from the reti testes is going to exit out of the testicle via efferent ductals. Remember our famous efferent means exit out of a gland of some sort. Okay, so that's why these right here, these three you're seeing, are the three efferent ductals that carry sperm away from the testes. And inside the efferent ductules, they're usually lined with cilia to produce fluid currents that will help propel the sperm along the way. And then they will finally get to the epididymis part, okay, which is this upper area. And we'll talk about different parts of the epididymis, okay? Now, if we want to zoom out of that, and so I can give you a bigger picture of this. So again, now you can clearly see this as in a bigger picture. So these here are the lobules. These here are the seminiferous tubules that's coiled around everywhere. This right here, the two that's right here, that's the straight tubule. And then this meshy coral looking thing is the reti testes. These three right here are your efferent ductals, and then they will get into the epididymis area. Okay? All right, so let's see if I can backtrack all that and go back into our presentation mode. Well, I'm just going to make that smaller and remove that. And then let me erase that. Okay. All right, so let's go back to our, I'll have to get a little bit smaller than that. Okay. All right, so let's go back to our route. So in between seminiferous tubules are what's called interstitial endocrine cell, what you were labeling or having to remember on the histological slide of testes. So those, I said, are those cells between the tubules that you're seeing here or here. Okay, so another name for these cells were Leydig cells, and they are important for producing androgens, which is, again, male sex hormone, and the main male sex hormone is testosterone. Now, once the sperm reach the epididymis, they will then travel through three different parts of the epididymis: the head, the body, and the tail. Now, the head is right here. This is the head of the epididymis. This area is the body of the epididymis, and this area right here is the tail of the epididymis. Now, it is in the tail of the epididymis that sperm are primarily being stored in while they're still trying to get towards the rest of the reproductive system. And then they will move on to the ductus deferens right here after they exit out of the tail part of the epididymis. Now, the epididymis tubules, okay, are lined with the pseudostratified columnar epithelium, epithelium, okay? And they have what's called stereocilia to increase surface area for absorption and secretion of fluid. Because a lot of the things that needs to happen with the sperm is that, yes, they can swim, but yet they need that fluid in order to propel them towards the outside of the body. They need to save the energy for later when they really do need to swim in the female reproductive system. So their main job within the epididymis is to monitor the composition of the sperm. Now, and they can recycle any damaged sperm by absorbing them back. So if there are any damaged sperm that can't swim or they look different or irregular, okay, they will be reabsorbed back into the body. They can store and facilitate maturation of the sperm for about 3 weeks before they are ready for excretion. Again, sperm should be able to swim by the time they arrive in this section of the reproductive system, but it's easier if there are fluid movement and peristalsis as well to help them propel them along. Okay, they need to save all their energy for the long journey within the female reproductive system. That, you know, I will upload a video to show you their process, their movement. Okay, all right, so the ductus deferens, or also called the vas deferens, is the duct that leads sperm from the epididymis toward the urethra. Now, before arriving at the urethra, there's actually an expanded region called the ampulla of ductus deferens. So, kind of like the duodenal ampulla that we talked about. So that's another good word to remember in your, sort of terminology list. If you see ampulla, the word ampulla somewhere, it usually refers to a small area that's expanded to temporarily store something before release. And that's what the duodenal ampulla was to temporarily store pancreatic secretions and bile before they're secreted into the small intestine. In this case, the ampulla of the ductus deferens is for temporarily storing the sperm before they're ready to be excreted out of the body. Okay, and within this area of the ampulla, the sperm can actually be stored for a couple of months. The ductus deferens is lined with pseudostratified ciliated columnar epithelium, which again, we have all those cilia there to help propel the sperm cells along by moving the fluid inside, and also there will be peristaltic contractions to help, move the sperm along. Okay, so the ampulla area, don't mistake it with seminal glands. So this is the vas deferens. Remember, the vas deferens comes from outside of the body via the spermatic cord. So from the testicle. So here's, for example, the testicle. Epididymis travels into the vas deferens, which then leads into the inside of the body via your inguinal canal. And then it will go towards the bladder. And then it will reach this ampulla region. So this is that expanded region we were talking about before entering the ejaculatory duct, okay, and the urethra, okay. So don't mistake seminary gland with ampulla of the ductus deferens, okay, especially when we're talking about the lab practical coming up. All right, so as I have mentioned in the urinary system lecture for males, the urethra is a passageway for both sperm and urine, and hence this is why males need multiple accessory glands for secretion that prevents the sperm from being damaged by the urine's acidity. Now, first of those glands that sperm will receive secretions from are the seminal glands. Okay, so this right here, this one and this one right here are your seminal glands. Okay, which is why this is the first gland that the sperm will hit when they arrive within the ejaculatory duct. So these are also called seminal vesicles, and they're for the purpose of producing the majority of the volume that makes up semen. Semen is basically the fluid that contains sperm cells. It is mainly alkaline in pH. Again, part of that is to help with the process of neutralizing any urine that's left over within the urethra. And then the sperm will move on to the ejaculatory duct to get into the prosthetic urethra. Okay, so this is the ejaculatory duct right there, and then this part is the prostatic urethra. Then we will receive another round of secretion, then the sperm will receive another round of secretion from the prostate gland, which is the next gland in the process. It produces prostatic fluid for the sperm that makes up about quarter of the volume of the semen, and they contain two components, important components. One is called the acid phosphatase and the other is amylase, both of which will prevent sperm coagulation within the female's vagina. Now, there will be some coagulation when the sperm is about to be expelled, but once sperm reaches the female reproductive system, then it needs to not coagulate because the sperm cells then need to come out of their, gelatinous layer and fluid and then start swimming around all the way to the fallopian tube. So if there's coagulation, then sperm will be stuck, okay, and they can't swim around, and that defeats the purpose of reproduction. So, and because once the sperm does enter the female reproductive system, again, they will need to swim up those fallopian tube. So that is why you cannot have coagulation once it enters into the female reproductive system, but they need to be coagulated before when ejaculation happens. And I'll talk about what enzymes and proteins are in charge of that in a bit. Now, there are quite a bit of problems for males that has to do with the prostate gland, however. For one thing, according to statistics, 1 in every 2 males will get prostate cancer sometime in their life. Fortunately, is not just by, you can actually detect prostate cancer via 2 ways, okay? Not just by manually feeling for the prostate gland. This is called the digital rectal exam, which is when the doctor will feel the size of the prostate gland through the anus. So that's already one good way to detect for enlarged prostate gland. Now, the other thing about cancer is that cancers are usually why some of the cancers are so aggressive, or why we can't detect their presence until later in stage, is because there are no markers within the blood that's circulating around the circulatory system. But luckily for prostate cancer, they do release something called the prostate specific antigen, or PSA, into the blood. So both by feeling of the doctor and by blood tests, males are able to detect for presence of prostate cancer pretty early, which is also explains why prostate cancer is one of the most high mortality, high survival rate of cancer, even though 1 in every 2 males will get it once in their lifetime, but they're also very much survivable. Okay, many males will survive through prostate cancer. Now, once males reach around 50s of age, they will have increased risk for another thing called benign prostatic hyperplasia. Okay, this pretty much mean a non-cancerous form of prostate gland enlargement. Because the prostate gland surrounds the urethra, that's why an enlarged prostate cannot be left untreated. Okay, it can go from the normal size of a walnut all the way to a huge size of a lemon. Okay, now the enlarged tissue, because it surrounds the urethra, all those extra tissue will then squeeze down on the urethra, which you can imagine this is the prostate gland right there, and it surrounds that part of the prostatic urethra. So when there's extra tissue putting pressure on the urethra, you can actually limit the ability for urine to pass, okay? And if it completely blocks out the urethra, then urine will back up into the kidneys, which can then lead to acute renal failure if the problem persists. So hence, usually enlarged prostate gland will be treated pretty soon, okay? And this is done via procedure called TURP, okay? TURP is right here, TURP. TURP stands for transurethral resection of the prostate gland. This is where a resectoscope, okay, it's kind of scary looking thing, but it's like a big, huge needle. It's actually inserted through the opening of the penis all the way down to the prostate urethra area to dissect away any extra tissues, and therefore they can remove the blockage and restore urine flow within the prostatic urethra. Okay, so the last gland that secretion is important for the fluids that surround sperm cell is the bulbourethral glands. So the bulbourethral glands are kind of tiny little guys right here. So you see these two dots right around the membranous urethra. That's a bulbourethral gland, which will secrete an alkaline mucus that will neutralize any acidity that's left due to urine being left within the urethra, because urine can be in that same passage as well, because they share the same passage, urine or sperm. There's actually millions of sperm cells per ejaculation, so some students would think, whoa, what's all the worry about protecting these sperm cells when there's so many of them? But for males that may have lower sperm count, sometimes everything matters in helping the sperm survive. So that's why you have these bulbourethral glands there to make sure they clear the pathway out of any acid from the urine before the sperm expels out of the body. Okay. All right, so as already mentioned, semen refers to both the fluid, which is seminal fluid, and the sperm cells themselves, okay? And the fluid contains secretions as contributed by the three glands we talked about, the seminal gland or seminal vesicle, the prostate gland, and the bulbourethral gland, and they each contribute a portion of the seminal fluid to the sperm. Now, there are also nerve cells within the seminiferous tubules, and those secreted within the epididymis that will contribute about 5% of those fluid. But as we have already said, most of the fluid in the semen is contributed by seminal glands, and we'll talk about what they contain. Now, the fluid from seminal glands supply quite a number of important things to sperm cells. They contain fructose, for example. Okay, fructose is a type of glucose, which means it is very important for sperm metabolism. Okay, remember I said they have a long journey to go once they enter into the female reproductive system. They have to sperm quite some ways. And the thing is, we'll talk about it in the next chapter, is that they actually swim against the current. It's kind of like salmon fishes swimming against the current to get to the top of the hill. Is kind of like that for sperm cells as well. So that's why they need all those sugar to metabolize into ATP, so they have the energy to propel themselves. Prostaglandin is also another important ingredient, which will stimulate smooth muscle contractions that should help with the peristalsis process of moving the sperm along. And then fibrinogen is what I was talking about, that temporary semen clots, okay? That temporary coagulation that has to happen when the sperm comes out of the male's body, okay? So that way it won't be sort of leaked out everywhere. Remember that clot, okay? Once sperm enters into the female reproductive system, if it's just a big spot of fluid, it can actually leak out of the reproductive system. And hence, to prevent that from happening, that's why you need fibrinogen there to form that temporary clot, so it stays within the vagina of the female reproductive system, and then they will be lysed into so they can further swim upwards, okay? But in the beginning, they need to be formed that clot with fibrinogen. And hence, the enzymes that is within the seminal fluids also has multitude of different functions. The protease within the seminal fluid will dissolve the mucus within the vagina. In the female reproductive system's vagina, there are a lot of mucus in there to repel microorganisms and trap microorganisms before they go up to infect the uterus. So that's why for sperm, they can be held tight within the mucus as well. The mucus is not very fluidy, so they can trap sperm, and hence that's why sperm needs protease to dissolve some of that mucus away. They also have another enzyme called seminoplasmin. Now, seminoplasmin is a type of antibiotic chemical, okay, that can actually prevent urinary tract infection for males. There's also prosthetic enzyme that will convert fibrinogen to fibrin. If you remember from our clotting lecture from the blood chapter, fibrinogen needs to turn into fibrin in order to form that clot. So same thing here for sperm. So prosthetic enzyme is what's going to convert that fibrinogen right here into fibrin. Now, kind of like once that clot is is already done this job in patching up blood vessels back in blood chapter, it needs to be removed. Same thing here. Okay, the whole point of coagulating the sperm is to keep the sperm within the female reproductive tract. Once it's in there, it needs to be lysed by fibrinolysin, which is another enzyme that will liquefy the clotted semen, so then now the sperm can freely swim up the reproductive tract. Okay, and last but not least, the sperm cells arrives at the penis, which is a tubular organ for reproductive and urinary system. Like I've said, it includes the root portion of the penis, the root, the body, and then the head. Okay, the head is actually named a fancy name, glans penis. Okay, which is the expanded region surrounding the external urethral orifice where urine comes out of or sperm comes out of. And for those that has kept their foreskin, okay, the foreskin is the medical terminology for that is prepuce. Okay. And preputial glands within the prepuce actually secrete something called smegma. Smegma is a waxy material that will prevent bacterial infection. So a lot of times there's this layer of skin for, if the male baby was not circumcised, they would form foreskin, which is the prepuce. But if males don't do a very good job of cleaning within that area, they can get infected. And hence that is why, to prevent infection, they do secrete that waxy material called smegma to prevent bacterial infection. But some males do still get bacterial infection pretty severely, and hence they need to do what's called circumcision. Okay, so hence to prevent that infection, a lot of times male babies are circumcised when they are born. Okay, probably usually a day or two after they're born, and this lowers the risk of bacterial infections, UTI, HIV infection, and even penile cancer. So, circumcision does a good thing. And for males, if they are circumcised as a baby, they will never remember that pain. But if you are circumcised at an older age, it is a very, very painful process to go through, especially during the healing stage. Okay. So the body of the penis contains 3 cylindrical columns of erectile tissue. 2 of those right here. So this is a cross section of the penis, and is enlarged here. These 2 purple circles represent the corpora cavernosa. Okay. Now, corpora cavernosa is the same thing as corpus cavernosum. Okay, corpus cavernosum is the singular version of corpora cavernosa. Okay, corpora cavernosa is plural version because males do have 2 of these corpora cavernosa, which surrounds a central artery right here, and that's why for a penis to erect, okay, they have to be filled with blood. Okay, we'll talk about that process and what nervous system is involved. And then you have the other erectile tissue called the corpus spongiosum, and that always surrounds the urethra right here. Okay, so the easiest way to remember which one is which, okay, is the one that surrounds the urethra is the corpus spongiosum. Okay, just remember, sponge surrounds urethra. Okay, the other 2 that surrounds the arteries for erection is the corpus cavernosum. Okay. All right. And the corpus spongiosum is what's going to end at the end right here to form your glans penis, okay, and also surrounds that last part of the urethra before the external urethral orifice. Okay, before we talk about the process of gametogenesis, which is the process of making gametes, we need to do a quick review of meiosis. Remember from Bio 1406 that you'll learn about the process of both mitosis and meiosis. Mitosis was the process of cell division for somatic cells, while meiosis is the process of how sex cells or gametes are formed. Now, mitosis generate daughter cells that are diploids. Okay, so remember they are diploid, which means they have all 46 chromosomes, and they contain the exact same genetic material as the parent cell. So they are nothing more but clones of the parental cell. That's what we do with our somatic cells like skin, intestinal epithelial cells, etc. Okay. Now, for meiosis, on the other hand, they will produce 4 haploid daughter cells. Remember, haploid, meaning they have 23 chromosomes, half the amount of chromosomes, and they are genetically unique or different from each other. There are 2 processes that contribute to the genetic variation. One is called crossing over, which happens within or during prophase 1 of meiosis 1. Now, crossing over happens between a pair of homologous chromosomes, where DNA is exchanged between the pair of homologous chromosomes. If you remember, homologous chromosome is 2 chromosomes that are of the same size and they carry the same genes within the same location. One you have inherited from your mom, so that's why one is called the maternal chromosome. And one you have inherited from your dad, which is called the paternal chromosome. So that's what the definition of homologous chromosome was. And they form form what's called a conformation called tetrads through a process called synapses. So the 2 homologous chromosomes actually lay on top of each other, and they're connected to each other through a protein lattice structure called the synaptonymal complex, which forms that synapses and creates this new conformation of the chromosomes called tetrad, meaning making up of 4 sister chromatids. Okay. The other source of genetic variation is during metaphase 1 of meiosis 1, where we can have different arrangement or alignment of homologous chromosome via what's called independent assortment. So maybe some of this will ring a bell from your what you learned in 1406. I'm not going to go into too detail here, but it's just a quick review of what meiosis is and what are the major points, and what's the difference between mitosis and meiosis. Okay, make sure you know the difference. Now, for meiosis, don't forget that there are two stages of cell division. Here's after cell division 1, you get 2 cells, and after the cell division 2, you get 4 cells. Okay? All right. So, hence, when males make sperm, the process is called spermatogenesis. Making, remember I said genesis means the making of, that's the root word, and the making of sperm. So the timing of this process is different from females, very different. Spermatogenesis only begins at puberty for males, and they will continue for the entire lifetime. It technically doesn't stop. The only thing that may change with age is the amount of sperm cells that are produced within the seminiferous tubules, and whether erectile tissues are functioning correctly in order to allow for ejaculation. So that's the only thing that's inhibiting or changing as a male gets older, but they will never stop making sperm. So the process of spermatogenesis goes from sperm stem cells, okay, so stem cells, and they are called spermatogonia. And spermatogonia are what resides within the seminiferous tubules, and they will differentiate into what's called primary spermatocyte. And at this point, they are still diploids containing all 46 chromosomes, and then they enter into meiosis. Okay, so that's why here we're still talking about mitosis or splitting of your stem cells, and then they go through meiosis to make sure they can halve their amount of chromosomes so you don't end up with aneuploidy or multiple copies or sets of chromosome within the embryo. All right, so meiosis is where your primary spermatocyte will become the secondary spermatocyte. Okay, so primary spermatocytes go through meiosis 1, meiosis 1 to become secondary spermatocyte, which will then further divide into immature spermatids. So this is the second step or second phase of meiosis. So secondary spermatocytes will go through meiosis 2, to become spermatids, which then half the amount of chromosomes into haploids. So up until secondary spermatocyte, they're still diploids, and then by the time they become spermatids, they are now haploids, containing only half the amount of chromosome. Notice that at spermatid stage, they still can't swim yet. You don't see any tails. Okay, that's why they need to become a mature sperm through another process called spermiogenesis. Okay, so from spermatids to motile sperm, okay, with the tail and everything, they have to go through spermiogenesis, okay, before they become these mature sperm. So once they become that mature sperm, then they can swim, become mobile, okay? They will also lose their attachment from nurse cell and enter into the lumen of the seminiferous tubule. So this right here is zooming in to one of those circular shape things I was talking about, the cross section of a seminiferous tubule we were looking at on the microscope, okay? And hence all of these cells you're seeing within the seminiferous tubules are these, okay? They go from spermatogonia, okay, to primary spermatocyte, to secondary spermatocyte, to spermatids, and then they will become sperm that will eventually leave out of the cytoplasm into the lumen. There is a lumen within the seminiferous tubules. Remember, that's why they're noodle shaped, okay? They're ducts, okay? So the lumen is where the sperm will then travel through all the coils and everything into the through the straight tubule, and then through the retitestes and the efferent ductals and onto the epididymis, okay? And then followed by the vas deferens. So that's how they are able to leave that area and sort of accumulate within the lumen and start swimming or be propelled by fluid movements within these ducts. Now, let's see. So for male sexual function to work properly, there are actually 3 E's, okay? Erection, emission, and ejaculation. Okay, so erection needs to happen first, where the parasympathetic stimulation needs to happen, where nitric oxide will be released to relax smooth muscles around blood vessels. Okay, so erection, contrary to what you think, is not contraction of muscles yet, okay? You need to allow the blood vessels within the corpus cavernosum to dilate and be filled with blood, and hence those tissues will be engorged and be able to erect, okay? So the parasympathetic stimulation is what allows the blood vessels within the cavernosum to dilate, and then blood flow will increase in that area, allowing for erection of penile tissues, okay? Emission is then stimulated by sympathetic nervous system. Okay, so sympathetic nervous system will increase the peristaltic contraction of sperm from the ampulla of the ductus deferens into the urethra. Remember, you have your bladder right here, and then the vas deferens coming in, okay, and then into that expanded region of the ampulla, and then they will merge to form your ejaculatory duct and then your prosthetic urethra. So for the sperm to be able to move from the ampulla region onto the urethral region, they need that sympathetic stimulation and contract the sperm into the urethra, okay? And the final ejaculation process is where then you will need somatic motor neurons from the inferior lumbar and sacral segments of your spinal cords, and that will cause contraction within the urethral tissues to push the semen out of the body. Okay, and this is why sometimes with lower spinal cord injuries, males can lose their ability to conduct ejaculation. Okay, because there are important neurons within that area that regulate this process. Okay. All right, so within the seminiferous tubules are very important cells that I've mentioned. They're called nurse cells, or another name for them is Sertoli cells. These cells are what's going to help the sperm cells to mature, leading them from spermatogonia all the way to become a spermatid and followed by a mature motile sperm. So their cytoplasm is where those spermatocytes will develop. So this is kind of like a section of that circular. Remember, we saw that circular seminiferous tubules, and I said there's these nurse cells all around, and including all the spermatogonia, all the developing sperm cells within that area, and then that's the lumen of the seminiferous tubule. So we're looking at a section of that within here, okay, to zoom in to what's happening within the nurse cell. So the nurse cell is actually huge, okay. All this purple area you're seeing is part of the cytoplasm of the nurse cells within the seminiferous tubule. These are nuclei of the nurse cells themselves. Okay, so the purple here is the cytoplasm. These guys are the nuclei of the nurse cell. And hence, all those spermatogonia, spermatocytes, primary, secondary, they are actually residing within the cytoplasm of the nurse cells themselves. Okay? Now, there's a blood-testis barrier as well, just as we have blood-brain barrier, blood thymus barrier, there is also a blood-testis barrier as well to separate the seminiferous tubules from general circulation so that we can protect the sperm cells from anything that has gotten into the systematic circulation because they're very important reproductive cells. Now, cytoplasm of the nurse cells is divided into two main compartments. The basal compartment, which is this right here, this dashed line right here, is what forms that blood-testis barrier. And below that blood-testis barrier is the basal compartment of the nurse cells. Okay, this part of the nurse cells is what contains your spermatogonia, which are your sperm stem cells. Okay, the luminal compartment, which is right here, okay, above the barrier line, that's where meiosis and spermatogenesis will occur as the spermatogonia becomes a primary spermatocyte, secondary spermatocyte, etc. Okay. Now, the other important role of nurse cells is to produce fluid in the lumen. Remember, the lumen is not just an empty space with air. There's actually fluid within there to push and propel the sperm along, okay? Or else the sperm will never get to their destination. They need fluid to be able to move. And hence, the nurse cells are in charge of making that fluid, okay? And the fluid usually also contains androgen, estrogens, potassium ions, and even amino acids. They also have immune system cells, okay, or actually, that blood-testis barrier is also functioning to prevent immune system cells from attacking the sperm, because technically speaking, these sperm are different genetically from the host, which is the parent, okay, or the male adult body. So if immune system was to see a sperm, they would actually think it's a foreign cell because you contain genes that are different from the rest of our body. So they will actually attack the sperm. So that's why the blood-testis barrier provides another important function to prevent, your body themselves attacking the sperm. Let's see. So the nurse cell's other functions are promote division of the spermatogonia, like I said, and meiotic division of the spermatocytes to become primary, secondary, and spermatids, and provide nutrients and chemical stimuli for the spermatid to mature and become a motile sperm. They also secrete a hormone called inhibin. Okay, remember we talked about inhibin all the way back from the beginning of the semester when we were talking about that endocrine system hormone chart. Okay, we talked about gonadotropin releasing hormone, GnRH, LH, FSH, and then down below we had one of them. Those was inhibin. Well, inhibin is a hormone that's important for serving as negative feedback, so that when there's too much sperm or too much of the sex hormone, there's always inhibin to come back and tell your hypothalamus and say, hey, we can stop releasing LH and FSH, and we can also stop releasing gonadotropin releasing hormones. So inhibin decreases the amount of FSH to tell it, hey, we have enough sperm, stop making that much sperm because we have nowhere for it to store them. So inhibin is a negative feedback to FSH and ultimately to GnRH to stop making going through the process of spermatogenesis. Okay. All right, so this is a quick spermatogenesis video that we can go through.
Sperm develop in the seminiferous tubules of the testes. Primordial sperm cells, called spermatogonia, lie dormant in the outer compartment of the tubules from the late fetal period until puberty. At puberty, the spermatogonia undergo mitotic divisions, giving rise to genetically identical cells. The cells grow gradually into primary spermatocytes, preparing to divide into cells half their size. Each of these primary spermatocytes undergoes a first meiotic division, forming two smaller haploid cells known as secondary spermatocytes. These secondary spermatocytes divide once more, giving rise to four haploid cells called spermatids, containing exactly half of the father's genetic material. Through a process known as spermiogenesis, the spermatids change shape, transforming into mature sperm cells. The spermatid's nucleus condenses, the acrosome appears, and the tail begins to grow. Much of the cytoplasm is shed. As spermiogenesis completes, the sperm cells break free from the Sertoli cell lining of the tubules into the lumina. The sperm cells are then stored in the epididymis, where they functionally mature.
So that wraps up, sort of give you an animation view of the whole spermatogenesis process. So that should help you envision how things are how things are happening within the seminiferous tubule. We will talk about the parts of the sperm in a bit, okay? But before that, let's talk about the hormonal regulation of the male reproductive system. Again, there's a whole hierarchy system where you begin producing hormones at the hypothalamus level, which is gonadotropin releasing hormone, which then will stimulate your pituitary gland to make follicle stimulating hormone, or the FSH. FSH will stimulate nurse cells, which nurse cells then will secrete androgen binding protein to increase the production of androgens, which is male sex hormone, and stimulate maturation of the spermatids to become mature motile sperm. Now, the other thing nurse cell that we just mentioned that they will secrete is another hormone called inhibin, such that when there are too much sperm around, when there are too much sperm around, then inhibin will be secreted as a negative feedback to the gonadotropin releasing hormone and the FSH to tell them, hey, we have enough sperm, let's not stimulate the nurse cells anymore for spermatogenesis, and we can stop, okay? The other route of gonadotropin releasing hormone is they will stimulate the pituitary gland to release another hormone called luteinizing hormone, or LH. Remember, I made a joke in the beginning of the year of the semester saying that we can easily remember FSH is for production of sperm because it spells like fish. Fish swims just like sperm. So that's why FSH was for production of sperm. LH is for production of the sex hormones. Okay, so LH will stimulate Leydig cell to secrete testosterone specifically. Okay, remember those Leydig cells that lies around the seminiferous tubules, and testosterone will do a couple of different things. They maintain libido, stimulate bone and muscle growth, maintain male secondary sex characteristics, maintain accessory glands and organs of the male reproductive system. Now, they will also negative feedback to GnRH if there is too much testosterone. Okay, so they're kind of different from how FSH work. To negative feedback on FSH, you need inhibin. Now, testosterone itself, being the product, will negative feedback back to your hypothalamus to stop the production of testosterone when there's too much of it. Okay, so make sure you understand these 3 different hormones or a couple different hormones for the male reproductive system. All right, so the sperm contains 3 main parts: the head, the middle piece, and the tail. Okay, so this is the head, this is the middle piece, and here is that tail. So the head is what contains that important 23 chromosomes that will contribute information of a baby from father. And at the very tip of the head are essential enzymes called acrosome, as mentioned in the video. So acrosome are what's important for the final process of, when the sperm reaches the egg, it is important for fertilization. Okay, it assists in the penetration of the sperm cell into the oocytes. Okay, and we'll talk about this whole process of penetration and how the sperm cell will finally merge with the oocytes in the next chapter. Okay, we'll talk about that process in detail. So the middle piece contains mainly mitochondria. Remember I talked about how the sperm's main job is to swim once it enters the female reproductive system, and it has a long journey to make from the vagina all the way up to the fallopian tube, swimming against the currents that's formed by the fallopian tubes, pushing the egg downwards toward the uterus. So that's why the sperm needs to have a lot of ATP around, and the way to make ATP is through mitochondria within the middle piece of the sperm, and they will metabolize that sugar that comes from the seminal fluid. Remember, the seminal fluid has fructose in it, and that's what they will use as a source of energy for swimming. Now, the tail of the sperm is the only flagellum that exists in humans. Okay, they move in a whip-like corkscrew motion. So a lot of people think they wig their little tail like that to swim, like swimming freestyle or froggy style. They actually don't move like that. If you've taken microbiology, flagella actually spins. Okay, so there's a little turbine back here after the middle piece before you get to the tail, and that turbine kind of shapes like cylindrical structure, and it spins at a very high speed, and when that spins, it actually spins the tail. So the tail ends up spinning to propel the sperm forward, kind of like how a propeller works to propel the boat forward. And that's what that spinning action is doing for the tail of or flagella of the sperm to propel it towards the oocytes. Now, the process called capacitation is when the sperm will become motile and fully functional. This is when, and this process is activated when it's mixed with seminal gland secretion. Okay, so that's the whole process of capacitation. Don't confuse it with spermiogenesis, however. During spermiogenesis is when sperm will lose all the other unnecessary cellular components because their main job is to deliver chromosomes to the female reproductive system into the egg cell itself. Okay, so once it has become this mature looking sperm cells, they don't really need ER or Golgi apparatus, lysosome, peroxisomes, all those other organelle that is just extra weight that the cell no longer needs. It's kind of like how the red blood cell loses its nucleus while it's trying to become a mature erythrocyte circulating around the body. Because for red blood cell, their only main job is to transport oxygen and carbon dioxide. Whereas for sperm is a similar scenario. They're just carrying the genetic material to the female reproductive system. So they don't need all those other extra weight on the cell and, make this, in a sense, make the cell swim a lot slower. So in order to make that swimming efficient for the sperm cells, all those other extra stuff will be removed via spermiogenesis, and capacitation is when then the sperm will become fully motile and fully functional. Okay, so let's take a break here and we will come back to talk about the female reproductive system in part 2 of the lecture video.